Hi, good evening, Ilkay. Hello, good evening.
Hi, Raman.
Hi, good evening.
How are you?
I'm good, very good. Not
Okay.
supporting. Things are okay in Turkey?
Yes, fine. Thank
Okay,
you.
here in India, it's going bad.
Okay.
Okay, nice to see you.
Nice to see you too.
So your
Thank you
slides are
for
working
coming.
fine. I'm happy. Slides are okay?
Yes, we were just trying it.
Fine.
How to
So
share.
5 o'clock will start?
Yes.
Okay, another 8 minutes. I'll just freshen up. Huh?
Sure. Okay.
Thank you, thank you.
Thank you. Bye bye.
Yapın hocam. Hiç o şey değil. Direkt. Ona bas. Ya orasına basmanıza gerek yok. Şu. Tamam.
Buna basacağım.
Tıklayın onun üstüne. Share deyin. Tıklayınca onu. Aynen.
Share diyeceğim.
Sonra
şuna geçeceğim. Şuna mı?
Evet. Sizin şeyiniz. Normal burada hani şey yapıyor.
Bir şey olmadı bunun. Ama ben onlar beni şuradan mı görecek?
Muhtemelen. Ya da oraya mı bağlanacak acaba?
Tekrar.
Yani iki kamera gibi bir şey. Ya sizi görmeyecekler.
Ya ben
Bunu
onları
şey
göreceğim. Ben
yapacaklar.
normal anlatacağım demek ki. O zaman tamam. Buradan halledeceğim.
Aynen.
Dur şuraya bir yazayım da.
Share
diyeceğim. Ondan sonra büyük.
İlk kutuyu seçip share diyorsunuz. PowerPoint'i başlatıyorsunuz şeyini. Presentation.
Tamam, inşallah bir şey çıkartmam.
Bekleyin.
Yok be.
Buçuktaysa.
Buçukta onların saatine göre 5.
5 dediniz işte.
2,5 saat varmış.
Ama şu an siz de kameranızı mı kapatsınız? İyi ve geçti. Stop video. En azından diğerleri gibi. Nasıl Türkçe konuşsak da anlamazlar.
25 kişi olmuş.
Ben hiç sona girmedim.
Ondan bir saatte yetiştirim inşallah.
3.30'da ne var? 4'te geliyor onlar. 4 çeyrek.
4 çeyrek falan demişler.
Tamam.
4 çeyrekte onlar işte gelir bir 15-20 dakika dururlarsa ben de 5 gibi çıkacağım. Öykü'yü havaalanına bırakacağız buradan.
İzmir'e gidiyorlar.
İzmir'e gidiyor 7.30'da.
Ya hocam eşinize potasyum klorür ampulleri var. Hani potasyum
klorür.
eksikliğinde meyve suyuna katıp veriyor hemşireler. Bize bayağı bir vermişti hemşire giderken. Şimdi babam diüretik kullanıyor ya. Bazen potasyumu falan düşmüş olur. Yani onu biz hani böyle şey yapıyoruz yani. Şey falan muz da yiyemiyor çünkü o da kabız yapıyor. Öyle bir şey getirebilir mi hastaneden acaba bir sorsanız?
Potasyum klorür.
Ampul yani çözeltisi böyle bildiğiniz ampulde.
Sorarım.
Bilir. Yani bilir hani normalde şeylerde de vardır da bizim hemşireler vermişti Onları bitirdik. Onu kırıp meyve suyuna döküyoruz.
İyiymiş valla.
Ama çok acı.
Vallahi.
Acayip yani. Bir galon şunlar böyle de. 2 ampul falan veriyorlar potasyumu düşünce onu. Öbürlerine infüzyon veriyorlar da.
Hatta hemen şimdi sorayım.
Başlar ya sonra sorabilirsiniz.
5 dakika vardır. Tabii burada açarsın. Gürdal ne yapıyorsun? Ben de şimdi konferansım başlayacak da diyecektim ya. Gökçen sordu da bu potasyum klorür ampulleri oluyormuş hastanelerde. Kırıp hastaların meyve suyuna falan dökülüyormuş. Ondan bulmam mümkün mü? Vermem. Hastaneden veriyorlarmış onu. Gökçe'nin babasına lazım da. Nasıl sana? Neyi atacak? Resmi mi? Resmi yokmuş.
Normal cam
ampullü oluyormuş ya hastanelerde potasyum klorür. Sorsana. Canım babasının potasyumu düşük işte. şey yapıyorlar, meyve suyun içine koyuyorlarmış. Hastaneden vermişler bol bol gazdan da bitmiş şimdi. Tamam. Hadi benim şey başlıyor. Hadi görüşürüz. Bay bay. Hiç anlamadım ne olduğunu. Sorayım dedim. Resmini göndersenize diyor.
Resim normal telefon. Onu kırınca at.
Allah. Ay Allah'ım ya. Sevmiyorum böyle online işleri ya.
Biraz evet hissetmek gerekiyor.
Evet
Gözlerin
ya,
gözleri görmek
hakikaten.
gerekiyor değil mi? Aha, millet geliyor.
Millet 42 kişi oldu birden.
İlk konuşma sizin mi?
Tek konuşma benim.
Ha, ben de onlar.
Rahime de mesaj atmış.
Bizim Rahime mi?
Ha,
Karşıdaki
Rahime
mi?
Şimşek.
Hı.
Benim Hindistan'da üyesi olduğum bir WhatsApp grubu var. Senin konuşman paylaşıldı. Çok gurur duydum falan diye.
Aa, nereden üyeymiş oraları?
Vallahi bilmiyorum ki. Orada diyor bir baktım paylaşıldı diyor senin için. Çok gurur duydum falan diye atmış bana görüntüsünü. Nereden nereye? 52 oldu. Anam. Bana hakikaten bu online hiç zevkli bir şey değil yani.
Ha, bütün katılımcılar görüyor. Ben yukarıda
Evet.
çıkanları şey zannettim.
Bunlar dinleyiciler, katılımcı.
Bunlar da konuşmacı gibi hani şey yapmışlar.
Ya benim bildiğim değil. Şu arkada benim şey niye görünmüyor?
İyiyiz.
Şu
Onu
büyütülmüş.
paylaşmadık ya.
O zaman şöyle dursun şimdilik. Ay. Kamala.
Restoran. El kaldırmış kamerayı. Ne istiyor ki?
Kayalı. Allah'ım hızlı hızlı anlatayım hemen.
Arkası ne ya şey gibi. Şu
Ya işte Yusuf Hoca da hep böyle yapıyor arkasını.
Demek ki Zoom'da var.
Varmış o. Bir de Mac'te oluyormuş bir tek. O gün
Ha.
Rümeysa soruyordu da şeyde. Siz çıktınız yani şeyden. Aman İpekler. Sonra dekanlar konseyi kalacağız ama arada bir 15-20 dakika herkes bir ayrıldı işte yemek yemek için falan. Bunlar geyik muhabbet yapıyorlar. Ben de kapattım her şeyi ama bunları duyuyorum. Nasıl oluyor? Şöyle mi oluyor? Zoom'un şurasından açıyorsun. Böyle yapıyorsun. Orada seçenekler varmış. İstediğini seçiyormuşsun. New York, San Francisco, Hawaii bilmem
Bu
ne.
evden bağlanıyor muhtemelen. Oğlu gibi küçük ya.
Ha, birileri giriyor içeri hakikaten. Millet de meraklı. Ben hiç böyle dinlemeyi de sevmiyorum. 61-62 şu anda. Bunlara sertifika da alacakmış da.
He, ondan
Katılımcı
belki
sertifikası.
kamera açıyorlardır falan filan. Normalde dinlersin sadece.
Live on YouTube. Ay, valla bitse de gitse bir an önce. Bir saat bir de ya.
Bir saat. Neyse geçer bir
Kaç
başlayınca.
slide? 64 slide'ım var.
İyi.
Anca yetiştiririm aslında hızlı hızlı gidersem.
Şeyde üstünde çizeceğiniz bir şey falan var mı? Slide şeyinde. Çünkü kalemle galiba olur orada.
Yok
Tamam,
sadece slide
o zaman
göstereceğim. Hatta bütün animasyonları da yok ettim. Çünkü İran'dakinde animasyonunu hazırlamıştım bir sürü. Aa, hepsi dondu. Yani tek gösteriyor. Ve üstüne getirmişsen bir şey mesela üstünde kalıyor. Sonradan gelmesi
kötü.
lazım ya
Anladım,
animasyonlar.
bir şey.
Bir baktım hiç oynamıyor animasyonlar. O yüzden bunu bütün animasyonları yok ettim ama sanki bunda çalışacak gibi.
Onda da mı böyle? Onda mı Zoom'daydı?
Onda başka bir şeydeydi.
Çünkü bu ekran paylaştığınız için ne varsa ekranda aslında
onlar
aynısı.
önceden istediler.
Onlar kendi yükleyip yaptılar.
Kendileri yüklüyorlardı. Bir de next diyordum. Ve böyle yarısı görünmüyor slide'ın, flu görünüyor. Ay çok fenaydı. Hadi Raman ya. Bunların da APTI diye bir dernekleri var ama çok büyük böyle bilmiyorum binlerce kişi. Onun da açılımı Association of Pharmacy Teachers of India. Hani Eczacılık Hocaları
gibi
Derneği gibi bir şey.
Mikrofonum
Mikrofonum açık. Duyuyorlarmış beni. O zaman şunu açayım.
Dr. İlkay, shall we start? With your permission?
Yes.
Looking as gracious as ever. So
Yeah.
enzymes, inhibitory natural products as promising frameworks in drug R&D.
Yes.
And the resource person for today is Professor Dr. Ilkay Erdogan Orpan. Am I right?
Yes, you are
I
right.
always call you Ilkay.
Okay.
Okay. She's the Dean of Faculty of Pharmacy, Gazi University, Ankara, Turkey. Born in 1972 in Ankara, Turkey. Did her BSc Pharmacy, Gazi University Faculty of Pharmacy, 1989 to 1993. MSc Pharmacognosy, Gazi University Institute of Health Sciences, Department of Pharmacognosy, Ankara, Turkey, 93 to 96, with scholarship provided by the Turkish Scientific and Technical Research Council. Wow. MSc Marine Natural Product Chemistry, and that also from Japan, 1996 to 1998. Okay, with scholarship provided by the Japanese Ministry of Education. PhD Pharmacognosy, Department of Pharmacognosy, Faculty of Pharmacy, Gazi University, Ankara, Turkey, 1999 to 2002. Postdoc, Fellow of Chemistry, University of Canada, 2003. Dean, Faculty of Pharmacy, now where she works. And I know that was in Cyprus, 2011 to 2014. Dean Faculty of Pharmacy, Gazi University, Ankara, Turkey, 2016 till date. And it's, I think, more than 10 to 15 years of association with Dr. Ilkay. She's the same person, and she has received many awards. And that is the Incitiment Award given by the Academy of Pharmacy Association of Turkish Pharmacists, Young Woman Scientist Award for Asia Pacific region, given by Organization for Women in Science for the Development of World, Italy. Science Award in Biology, that also is given by Islamic Corporation. Young Female Scientist Award, again given by the Turkish Academy of Sciences. Honor Award represented by Gazi University. Innovation Award for Women by Turkish Scientific and Technical Research Council and Association of Women and Democracy. Then Science Award, then Achievement Award, then there is a Health Award, there's a Golden Award, and one award from Me Too, the Best Human Being Award. Okay, publications, 237 articles in international journals covered by SCI, 43 articles in national journals, and 16 book chapters, 2 books, H-index, 44.3, patents 3, 4 patents application filed. She is member of Traditional Chinese Medicine Group in European Pharmacopoeia Commission, member of International Scientific Board of Austria Drug Screening Institute, member of Gazi Best Board of Gazi University, representative of Southeast Europe and Turkey for Phytochemical Society of Europe, and research interests mainly are phytotherapy, enzyme inhibitors from natural sources, neurodegenerative diseases, marine natural products, natural cosmetics. Most gracious, responsive, wonderful person, and of course, my international sister.
Thank you.
APTI is honored to have you on our forum. The session is all yours, Dr. Ilkay. Please.
Thank you, Raman. Thank you very much for this long and very kind introduction. And it is a big honor to be with you, with the participants here today. So I want to share my slides with you now. Okay?
Yes.
Shall we start? Okay.
Yeah, please.
So can you see my slides properly?
Yes, we can.
Yes?
Yes.
Great.
Perfect.
Okay, so I'll start. Okay. Raman, thank you very much again for this very nice introduction. And I really wish to be with you guys face to face, but of course now we are seeing each other from overseas, but it's good to be, chatting through online as well. So within this, I think next 1 hour, I will try to cover the subject related to enzyme inhibition of natural products. How can we use them as a promising frameworks in drug R&D? Actually, this is my major topic in research, but when I start, I will give some examples from the common research, and then I will do my best to give some examples from my own work. So, of course, I think most of you are familiar with pharmacognosy or natural product chemistry, because this is a very, very important subject for drug R&D. Natural products, as you can see in this slide, it is like an ocean, and we as scientists are like kind of divers to fish these beautiful bioactive molecules from, you know, either plants, animals, microorganisms, or marine organisms. These kind of, you know, bio resources could be a starting material for this kind of research for all of us, actually. So if you are lucky, you will, you know, catch up a very, very good or promising compound from these biological sources. So, of course, as I mentioned, you know, animals rather than, I mean, plants, animals, marine species, they are also very rich in, you know, chemicals, which could be later developed into drug candidates. So poisons or venoms, as we called, produced by these creatures, by these animals, can become you know, drug candidates. So there are some examples, as you can see in the slide, like captopril, which is an ACE inhibitor used for, you know, treatment of hypertension. Acyclovir, this is a DNA polymerase inhibitor. Initially, actually, I will also explain it a little bit later, but it was actually isolated initially from a Caribbean marine sponge. There is another interesting compound which you might be familiar with, like zyconotide. This is also calcium channel blocker, isolated from the venom of Caunus magus. This is a marine creature as well. And another compound, exenatidine. This is also a very promising drug. Actually has become drug as a derivative. So it is a GLP-1 analog isolated from the venom of Gila monster, which is Heloderma suspectum. So this work is quite joyful, quite attracting, because the first two compounds that I've just mentioned actually brought Nobel Prize to their discoverers. So it is also fun to dream, to have a Nobel Prize from these sources as a scientist. Of course, it's a big dream for all of us. And another source, microorganisms. Of course, we know that they produce very beautiful compounds. Usually they are antibiotics, but not only antibiotics, also some other drugs like acarbose, which was isolated from a fungus species, Actinopylanes. This compound is alpha amylase inhibitor, and now it is in the pharmacies as a diabetes drug. And marine bacteria, they're also very important, and most of them actually are thought to be, symbiotically lived with some sponges or other marine organisms. So they can produce very, very promising, nice, auspicious compounds like, MAO inhibitors, I don't know, ACE inhibitors, protease inhibitors. So we could get a lot of bioactive compounds from these kind of bacteria as well. And another fungus like Aspergillus terreus, that one produced a HMG-CoA reductase inhibitor, which is called lovastatin. It is also the start for discovery of statins, which is used for treatment of hypercholesterolemia. So I just mentioned about this creature. This is Heloderma suspectum, also known as Gila monster, and the venom of this creature actually afforded a very nice molecule, a very complex molecule actually. This is called exanthem 4. It is a protein derivative, and it was, by chance discovered that it has a very potent anti-diabetic activity. And the compound that I was talking about, is a synthetic derivative of this protein called exanthem 4, and now it is also in the pharmacies in many countries as an anti-diabetic drug. You can here see the chemical structures of this drug or molecule, and you can see how complex and how big it is. Actually, this has also some privilege, like this is the first one of this kind of drugs which is used for treatment of type 2 diabetes, and this class of drugs is called incretin mimetics. And another one that I just mentioned, zyconotide. This was isolated from a marine snail, actually. This is called Conus tessulatus. So this creature or marine animal lives in the ocean, in the Indian Pacific Ocean as well. And its venom, actually, it's a toxic or venomous animal, and it produces a series of toxic compounds called conotoxins. These are neurotoxic peptides, and they have ability to modulate the activity of ion channels. So this compound, zyconotide, produced by inspired by a molecule produced by this organism, is actually a synthetic molecule with a very, very potent analgesic effect. And in 2004, it got FDA approval, and now it is also used as a drug in clinic called Prialt. So anyway, of course, we can give a lot of examples of drug molecules already used in clinic, inspired by the natural products or natural compounds or molecules, whatever you say. So it is important to search our biological source, mostly, of course, plants, and then animals, and then microorganisms. Of course, all of them are like immense sources of drugs molecules, which still waits for us to be discovered. So coming to my own work that I would like to mention mostly actually in this seminar. So our workflow is, as you see in this slide, like, of course, each country like India, China, or many countries, Kenya, Turkey, Europe, they have their own traditional or folk medicinal data. And we have all people actually would like to use traditional knowledge or traditionally used plants for their health. So traditional medicine, or we call folk medicine, is quite important. And in India, I know you have this beautiful, and this is very impressive traditional medicinal system called Ayurveda. This is very important and a very, very nice source for not only for India, but for all human beings. So what we do, actually, we sometimes rely on this traditional medicine, the medicine data, or sometimes we do random screening, especially with the plants. We just collect the plant that we target, and we extract, of course, and then we do primary screening, or pre-screening we call. And in my lab, we usually apply in vitro methods for this. We use ELISA microplate reader to set up the enzyme inhibition assays. So I will just give information about those enzymes that are available in my lab. And then we just get active extracts. And then from the active extracts, we choose one or two, whatever. Then we, perform activity-guided fractionation and isolation, of course. And in each step, we do activity-guided fractionations. And from active fractions, of course, our final target is to identify the active compounds, or which compounds are responsible for that mentioned activity of the extract. Usually, all of us try to publish our data in nice reputed journals, and they always ask, when you submit a paper on extract, most of the journals or reviewers, they ask you what is the compound or compounds responsible for the activity of this extract. So for this purpose, you know, we do some phytochemical analysis. Sometimes we do this time-taking, time-consuming chromatographic procedure. Sometimes we do LC-MS analysis, and when we identify the compounds responsible for the activity, and then we of course elucidate the structure, chemical structures, and then we proceed them to the molecular docking assays. So this is usual workflow that we applied in my lab. So as you see the topic of my talk, of course, I'm doing enzyme inhibition studies on natural products. And for this purpose, we have a number of enzyme inhibition assays available in my lab. These are like cholinesterase family, phosphodiesterase 1, carbonic anhydrase 2, lipoxygenase, xanthine oxidase, very recently HMG-CoA reductase, like cytidine type of compounds we try to discover, and then tyrosinase, elastase, and collagenase. Thus, three enzymes that I mentioned are actually related to mostly cosmetic research. So we also do cosmetic research as well. So I will also give one example about that. And when you look at my background, actually my paper, most of my papers are related to neuroprotective activity of natural products through enzyme inhibition mechanism. So when we look at this activity among natural products, we see that usually phenolic compounds are quite active. Of course, that doesn't mean that only phenolic compounds show neuroprotective activity. No, that's not true. Actually, many type of compounds like alkaloids, saponins, coumarins, maybe a kind of subclass of phenolics we could consider. So anyway, many type of natural products actually show this activity, as you know. So, but phenolic compounds are a very large group among secondary metabolites of in the plant kingdom. So that's why I'm also particularly interested in phenolic compounds, and considering like neurodegenerative diseases like Alzheimer's disease, and we could see that phenolic compounds are quite dominant, acting through different mechanisms, as you can see here. So if since we have a limited time, so I skip those details. But what we do as a research relevant to neuroprotective activity of natural products. Actually, as I mentioned, we do in vitro methods like tyrosinase and cholinesterase inhibition. Of course, cholinesterase inhibition is very well known for the inhibitors which are effective against Alzheimer's disease. And tyrosinase, I just mentioned that it is connected to cosmetic research for skin whitening effect. Its inhibition is important in that way, but lately it has become a target for Parkinson's disease as well. So that's why I include this enzyme in neuroprotective activity of natural products as well. So we have in some in vivo methods, like we use mice using passive avoidant test, and also, as you know, Drosophila melanogaster. This is a kind of fly. So we use the brain homogenate of this fly, and you know, try to measure anti-aging effect of the compound or extract, whatever. And then as I say, we proceed these compounds, active compounds, to in silico analysis, applying some molecular docking experiments. I will give examples. And lately, actually, very lately, within this year, last year, we tried to collaborate with some groups to do cell culture assays in a relevant way. So I will start giving some examples from my own work. So as I said, I'm very much interested in phenolic compounds, which is like an umbrella group, and under it there are lots of subclasses, and chalcones and flavonoids. Of course, they are a large group as well. So this was a collaborative work between my group and a group from Institute of Soil Science and Plant Cultivation, which is located in Puławy in Poland. So they are very good in isolation of these natural molecules, and in this special work they isolated many flavonoid and chalcone derivatives actually from the female cones extract of polyshopes, which is Humulus lupulus. This is a very famous phytotherapeutic plant, as you know, and here you can see a lot of derivatives of these compounds, and here you can see some promising ones. You can see the IC50 values as compared to that of galantamine. You can see here on your right, on the bottom. So we could get some promising, chalcone or flavonoid derivatives from these plants as cholinesterase inhibitors, especially these two ones. So we do ATME analysis of these compounds as well. There are lots of parameters for this, and here you can see. And binding energy is, of course, important, especially for in silico analysis of these compounds. So through the binding energies, we can just predict how strong the interaction between the ligand, which is our molecule, and the active side of enzyme, as you can see here. Here you can see the compounds that I just mentioned, isolated from Humulus lupulus. You can see here some weaves of these compounds in interactions of active gorge of cholinesterase enzymes and these ligands. And by some colors, I try to indicate interactions, like by pinkish color, you can see the hydrogen bondings, and with dark green colors, you can see the pp stackings between the compound and the enzyme. And of course, with light green colors, you can see the hydrophobic interactions between these ligands and the amino acids located in the active gorge of the enzyme. And here, another view with the same compounds in terms of 3D appearance. And this seems more complicated maybe, but here you can see the clouds between the compounds, which shows electron densities of these ligands or compounds. So still we can see clearly how these compounds act or interact actually with the enzymes located in the active gorge. So this was the work, the collaborative work between this Polish institute and us, which was published 2 years ago in Phytomedicine. Another work that I would like to mention is about the compounds isolated from Bellamdecamna chinensis. This was also a collaborative work between my lab and another group from Wrocław University in Poland. So in this work, the group from Poland actually isolated a number of isoflavones and stilbene derivatives from Iris domestica, which is also known as blackberry lily, and some actually very well-known compounds came out. So we tested all of them against this cholinesterase family, and here you can see some moderately inhibiting compounds, like very well-known resveratrol and irulan B, piceatanol. It's another stilbene derivative. It is not that always you get very promising, very active compounds. Of course, sometimes you happen to find compounds with no activity or with very low or moderate activity, as you can see here. But with the cholinesterase inhibitory activity, especially with piceatanol, we could observe some promising inhibition I would like to mention. So with these three compounds, which seem to be moderately active, here you can see the interactions, actually had the hydrogen bondings between these compounds and the amino acid located in the active side of these two enzymes. You can see here. And this one, with the Polish group, actually we published in Phytochemistry Letters last year. Then again, a collaborative work with the phenolics from Yuka Shidigera. So actually the plant, although we did the work, joint work with the Polish Institute in Puławy that I just mentioned, the plant actually comes from Ukraine. It's a Ukrainian plant. So this Yucca species, probably in India, you have a lot of species of this genus anyway. So many or a number of compounds were isolated from this Yucca species. Most of them were actually known compounds, but when we tested against these two enzymes, cholinesterase family enzymes actually, you can see either very low inhibition or maybe very few moderate inhibitors among these compounds. So they're not promising, but anyway, activity is activity. And more compounds from Yucca shidigera, Yucca derivatives. Actually, these ones that you see in this slide are new compounds. That's why it took a lot of time to identify them. And when we tested against these cholinesterases, we could see more compounds with higher promising inhibition, I would say. Here see the IC50 values, and especially UCAL-B, it was the most active one. And this one, this paper actually came out last year with, as a joint work actually in molecules. So with the same Polish group, so we still go on collaborating. And this plant, Herniara glabra, this is a Polish plant. And this group from Poland, they isolated a lot of saponins again. And you can see all these three compounds actually are new compounds, new saponin derivatives. And again, we tested them against a number of enzymes, including cholinesterases, xanthine oxidase, elastase, collagenase, tyrosinase. And with these 6 kind of enzymes, actually, these saponins didn't show any inhibition or, you know, very, very low inhibition. But of course, we also know that enzyme inhibition is not always the mechanism for a compound to show any activity. So you also need to try different experimental methods to identify such an activity, because it might not be active through enzyme inhibition, but some other mechanisms. And if you have a chance, if you have an opportunity to test them in another experimental method, then you should try. So anyway, for this purpose, we also collaborate with another group from Turkey, and we now we have been testing these compounds in neuroblastoma cells, although they are not active in enzyme inhibition tests. We might get some nice data, but it is still going on, so that's why I'm not able to give or present the data here. But with the enzyme inhibition part of these compounds, we just published the data in Phytochemistry. It's a very, very fresh paper of this year. So as I said, coumarins, these are also very interesting natural products. I'm also interested in coumarins very much because, based on my experience, I have witnessed a lot of coumarins with really nice inhibitory activity, a selective inhibition actually against butyrylcholinesterase rather than acetyl derivative. So here you can see a lot of papers or nice review papers on coumarins, how they're acting on, you know, central nervous system. I had also 2 review papers on this kind of activity of coumarins. So anyway, coumarins are quite bright compounds in my experience. So I will give some examples of my work with the coumarins. So here you can see some coumarins isolated from different plants like Angelica officinalis and Mutellina purpurea. This was also joint work, still going on actually, joint work with the Faculty of Pharmacy of Medical University of Lublin. So my colleagues over there, they're very, what do you call it? They're very good in isolating coumarins from different plants. So they keep isolating these coumarins, like very well-known ones, Imperatorin, Xanthotoxin, Pterixin, and Huygens, and more you will see. So we started with those four compounds, and we published in nice journal like Food and Chemical Toxicology, Phytochemistry, and etc. And these are the data we have just finished, and they have not been published yet. We still prepare the manuscript. But anyway, as I mentioned, this coumarins from Heptapteris silicica, which is a Polish plant. Some known coumarins came out from this plant. And here you can see when we tested them against cholinesterase family again, we could see some promising one like colladonin and acetoxybadrakemin, caratovicinol, which is an interesting type of coumarin actually. Here you can see more coumarins isolated by the same group from another plant called Neocryptodiscus papillaris, more known coumarins, and among them actually they were like moderate inhibitors, and oxypusedanin, it was a promising one like isoimperatorin and ostol. Of course, the active ones are always proceeded to in silico experiments through our work. So more coumarins, actually 3 more coumarins from another plant, Petroedmontia syriaca, marmicin, daltoin, and simirnoirdin. Among them, only daltoin showed some moderate activity, which is in progress to prepare the paper. So another coumarin work that I would like to mention you is about semi-synthetic coumarins, because natural products are important not only because they're pure molecules from natural sources, also they are very good models of or frameworks, as I underlined in my title. They could be a very good frameworks for drug development, research and development. So that's why their synthetic or semi-synthetic analogs or congeners are also important for this kind of research. And these semi-synthetic coumarins, almost about 30, close to 30 in number, was a joint work with University of Chieti in Italy. So our Italian colleagues, they semi-synthesized this coumarin, so we tested them. And here for each coumarin derivative, you can see the isopeptide data calculated for butyrylcholine esterase. So, and especially one of them, the first one actually in the red frame, you could see it's very active against the butyrylcholine esterase, much more active than the reference drug galantamine. And this is ATME analysis of the compounds, as you can see. And this is binding energies. We can see the binding energies, how, what do you call it, how it is important for interactions of these ligands or compounds to interact with the active side of the enzymes. Here you can see the two-dimensional weaves of these active compounds, especially the first one. You can see most of these compounds have selective activity against butyrylcholine esterase, and here for the same ligands or compounds, you can see the three-dimensional weaves of these compounds with the butyrylcholine esterase. So all these actually data just emphasize that for this kind of compounds, of course, presence of a deoxygenated geranyl chain seems to be compulsory for inhibition of this butyrylcholine esterase. So this work came out last year in bioorganic chemistry as a joint work. And with the Wrocław Medical University, this was another work, a joint work that we performed. So tanchinones, that you must have heard about these compounds. You know, tanchinones are very famous or reputed diterpene derivatives, firstly isolated from danshan. You know, Salvia meteoris, a Chinese sage, famous sage species. So anyway, the similar type of tanchinones were also isolated from this plant, Perovskia atripelicifolia. You can see this is also called Russian sage. From the roots, these compounds, these tanchinone derivatives were isolated and they were tested again by us against this cholinesterase family. One compound, isograndipoliol, this was a new for this species, not new for the nature, but for this species. It was a new compound and it had a very promising, actually, a cholinesterase inhibitory activity. And here you can see the hydrogen bondings with the compound or ligand with the enzyme. And here you can see more hydrogen bondings in yellow color. Actually, you can see, especially with the residues or amino acid residues actually located in catalytic tride and peripheral anionic side of this enzyme, these two enzymes actually. And this was also published in Phytochemistry in 2017. So of course, I can give a lot of examples of these kind of work. I have a lot of papers, more than 200 papers related to enzyme inhibition. But of course, as I mentioned, we also do some in vivo experiments. This passive avoidance test done in mice is available in my lab, but we don't use it very often, to be honest. So anyway, but it is available in my labs. I would like to mention an in vivo study related to neuroprotective activity of natural products, actually. This was an interesting work and actually very fresh work. The essential oil producing company in Turkey, you know Antalya, it's a very famous touristical city in Turkey, of course, and the company is located in Antalya, and the name of the company is Talya. They produce a lot of essential oils and they export. They obtained essential oils from the plants growing in Turkey, especially in the Mediterranean region. So this Talya company, actually, they formulated essential oil mix for what purpose? It is formulated for motivation and enhancing mood, especially improving the memory and so on. Just directed to or targeted brain or memory enhancing effect. So here you can see the plants or essential oil of the plants in the formula. Of course, this is a patented formula. That's why I didn't give the species name of the plants, but here you can see some very well-known species from salvia, lavandula, lavender, rosemary, and two citrus species. And in this essential oil mix, when we analyzed by GCMS, we found that limonene was the major compound constituting like over 90%. So this is a very major compound in this formulation. So we tested in vitro for its esterase inhibitory effect and in vivo test in collaboration with the colleagues from Romania, University of Iași. So in the in vivo part, of course, the Wistar adult male rats were used, and you can see this MO, the essential oil mix that we name it like this. And in different in vivo tests, like NOR test, null object recognition test, or radial arm or Y maze test, in against amnesia induced by scopolamine. So here you can see the graph of these experiments. I'm not going to go into detail much, but here you can see with this essential oil mix called MO, in all tests, actually, we had very, very nice and good results with this essential oil mix. So it improved, novel object recognition memory. So it also it was able to improve memory performance in the Y maze or radial arm maze test. It was also good in improving spatial memory in the maze test, and this essential oil mix patented formula exhibited a good anticholinesterase effect, and also it was able to enhance the activity of the antioxidant situation of brain through this antioxidant enzyme, like superoxide dismutase, catalase, and so on. So since we know that the limonene was the major compound in the essential oil mix, so we also docked limonene against butyrylcholinesterase, because as you can see, it had higher inhibitory potential against butyrylcholinesterase. And here you can see the interactions with these compounds. And this is a very fresh paper that we published in Molecules this year, maybe 1 month ago. I think 1 or 2 months ago it was published. So coming to second part of my talk related to cosmetic research, of course we do tyrosinase, elastase, and collagenase inhibition assays for skin aging. To produce some, anti-aging products, as well as skin whitening or skin bleaching formulations, maybe I could say. So within the PhD work of one of my students, so we screened actually over 100 plants and did activity-guided fractionations to be able to produce some cosmetic formulations. So what we have done, one of the most effective plants was Cotinus cogrea. It is called European smoke tree. It is also grown in Turkey widely, but as a not as a medicinal plant, as an ornamental plant. You can see the appearance of this small tree actually in the pictures. So we used leaves and this smoke-looking parts or flowers of the flower extracts of this plant. And here you can see a time-consuming chromatographic analysis, but this is based on activity-guided fractionation and isolation, and each step of this chromatographic procedure. So we tested each fraction separately against these 3 enzymes, which are tyrosinase, elastase, and collagenase. As you can see, more fractionation by column chromatography and some very promising fractions against these enzymes. And this is also fractionation, chromatographic fractionation scheme of the pedicel part of the flower. As you can see, again, the same enzyme inhibition assays at each step of this fractionation through the chromatographic procedure. And here you can see, after all this, tiring and boring chromatographic experiments, so we were able to identify these 4 compounds in the active fractions. So these compounds actually came out as the now compounds, like 2 quercetin derivative, 1 camphorol derivative, and gallic acid metal ester. So we tested them against enzyme. We did in silico analysis as well. And here you can see this was also accepted very, very recently, maybe 2 weeks ago, and it came out this year as a paper. But of course, cosmetic research is fun. I mean, it is really enjoyable. And through this press screening, we were able to find more and more active plant extracts, actually, like, you know, grapeseed extracts, green tea extracts. These two are used a lot in cosmetic products. And another two, which is Cotinus cogrea, the European smoke tree, as I just mentioned. Another one is Maclura pomifera. This is called Osage orange. This Maclura is native to North America, but is also grown in Turkey as an ornamental tree. So from these 4 plants, we designed some formulas in 2 forms. One is microemulsion, another one is vanishing cream. So we created a brand name called Vitabel. But of course it has not been commercialized. So these are like prototype products. So we got some awards with these formulations. For the Cotinus cogrea, actually, the anti-aging formulation that we designed, we carry out some stability tests. In this sense, we collaborated with a cosmetic company in Turkey called Vivo. They actually function both in Turkey and UK. So anyway, they helped us to do all these tests for these formulations. And here we did particle size measurements in our faculty, actually, especially for the microemulsion type of formulations. Here you can see the examples from the grapeseed extracts and green tea extracts. And with some citrus species, actually, we prepared a hand cream to get rid of these, you know, dark spots. Usually happens in our hands, in our skin anyway. It has some skin whitening effects that we found in our press screening work. So anyway, we designed these formulas as a hand cream for the citrus ones. So we got some prizes. This is my PhD student, Cesar, and Raman knows her very well. She's a very hardworking person. So anyway, it was a part of her PhD thesis like 4 years ago. So anyway, we got some awards for this kind of cosmetic work. And almost coming to the end, I would like to mention about this work as well. This was started like 3 years ago. So my colleague from Hacettepe University, also a nice university located in Ankara. So she is a pharmaceutical chemist, and she keeps, you know, synthesizing the derivatives of alpha-kojic acid, allomaltol, and chlorokojic acid. So alpha-kojic acid, as you may know, actually it is a natural metabolite as well, derived from Acetobacter and Aspergillus penicillium. So this compound, kojic acid, actually is used as a reference for tyrosinase inhibition, for especially also it is an ingredient in skin whitening products. So anyway, she and actually her team synthesized over 100 derivatives of these 3 compounds, starting from these compounds actually. So we got some hits when we tested against tyrosinase. And of course, I'm not able to give all these derivatives, but anyway, especially among these 125 derivatives, there were, of course, effective ones, a lot, but one of them, a single compound, was quite active. So we decided to patent it. So we applied to Turkish Patent Institute and we got the patent. And my friend who developed this, who synthesized these derivatives, she also established a company called Cosmetic. In 2018, we got, 2017 actually, we got Turkish patent. And 2018, we got United States patent for this compound. And this is the team who worked on these derivatives, kojic acid and allyl maltol and chlorokojic acid derivatives. So we got the silver medal 3 years ago for this work. And last year in 2019, we got European patent for that. And then in 2018, again, as this woman team, actually, we got best academic invention medal in Istanbul International Inventions Fair, which we're proud. So coming to the end, actually, so I will just try to give examples from my own work, how natural products could be developed into drug candidates, how they're important. Of course, we know how they're important, but anyway, to underline that, you know, they could be used as direct molecules for treatment of different diseases threatening human health. And nowadays, as you know, whole world, all of us are, you know, dealing or trying to combat with COVID-19 or new type of coronavirus. And this coronavirus chaos also showed us how could be, how natural products could be actually. So anyway, we do in vitro methods, I mentioned, and some in vivo methods and in silico methods. And we have just started cell culture assays in collaboration with some groups in Turkey. So it is a very complex research as well. It is not like one direction. You need to know, you need to try a lot of mechanisms to identify one active, promising molecule, but they're inspiring. So I really like this type of research, and I'm really glad that I'm into this research. This is pictures from my small group, and of course I need to acknowledge the friends or colleagues that I've been collaborating through this research. I'm really grateful to them. And international collaboration is also very important to develop our research into global science, you know, not only national science, but global science. We have to share our power. We should come together to develop better papers, to find better bioactive molecules anyway. So in the end of this talk, actually almost finished, and I'm good in timing, I think. So these are some pictures from my university. Gazi University is a top leading institutes in Turkey, in top 10. We are also research-based university and Faculty of Pharmacy, which I am the dean, is one of the reputed or oldest pharmacy faculties in Turkey, founded in 1968. So I'm really, really grateful to you for listening to me, even online. Thank you very much for your attention.
Okay. Thank you, Dr. İlkay. I think it's around 3 o'clock at Ankara.
No, 3:30 almost.
Okay, so long time since I've come to Turkey, that means.
Yes.
So, for the interest of the participants, I would like to delightfully tell that me and Dr. İlkay not only have the common research interest, but we have been a number of beautiful places at Turkey.
Yes. Sure.
Of course, the unforgettable Kilis tape.
Yes, of course.
We did each and every place together, and she shopped like mad.
Yeah.
Also, where she studied and place where she stayed in her childhood.
Thank you.
We had baklava together, Turkish delight.
Yes, yes, of course.
So, Turkish tea.
Oh, yes, yes, yes, yes.
Lovely. I still have the cups which you got for me.
Okay, good.
Thank you, Dr. İlkay. It was a session beyond compare. Your slides, as always, were commendable, remarkable. appreciate. And APTI expresses its gratitude to you for agreeing to deliver the lecture for our teacher friends here in India. The session for questions. Kindly participants raise your hand if you have any questions. I will unmute you. Unmute otherwise. So here we go.
Yes.
Okay, Dr. Kamla.
Yes.
Yes.
Hi, everyone, and Dr. Ilkay.
Yes.
I am very happy to meet you here. Maybe you remember me. I'm from Azerbaijan Medical University, and
Okay.
we are friends in Facebook too. I was
Yes.
I was in your university during my visit to Ankara, during my visit to the
Okay.
conference in the
Oh, yes,
university.
yes, yes.
Thank you very much for so informative and helpful presentation, and thank you for organizers of this event, of this webinar. Thank you very much. Thank you
Thanks,
very much.
Raman.
And my colleagues also here from the faculty, because I work at the medical university at the pharmacy faculty, general and clinical chemistry. And my colleagues here, maybe they have some questions.
Okay, go ahead.
If you can, they haven't some chat place, but where
Yes.
they can write their question, Mr. Rahman.
Sorry.
Because some colleagues have some questions, but here you haven't some place where they can write the question and for the voice.
Ah, so Raman can do it.
Yes, chat place.
Can you just read the questions?
Because many a time the participants tend to spoil the slides, so we have disabled the annotations. You can ask your.
I don't know, my colleagues. Have you questions or not? So
You can just ask their questions. Because I don't want to touch the screen. I don't want to screw it up. So just ask whatever they ask.
I saw here some slide with nanoparticles. Maybe you work with nanoparticles with nanotechnology field or not. But how your work related to the nanotechnology? For me, it is because my search field, one of the search fields, this is nanoparticles.
I see. Actually, I don't know, Kamala, how you saw those nanoparticles, because I have nothing to do with nanoparticles. I have no work with the nanoparticles.
Just in one slide,
maybe in microemulsion part, you know, we had also some nanoparticles in this microemulsion formula in a nanoparticle size. But actually, it is not my own research area. So, you know, I don't work with nanoparticles, nanotechnology. I wish, but not yet.
Inshallah.
Inshallah.
Thank you. Thank you for your question. If you have some more, we'll get back to you. Now we have the top researcher from India, Dr. Vandana Patwale.
Yeah, Kayala Mamadova and Ayten Shkirova from my university. They have some questions. If you can, please. Can you open their voice?
Me?
No, not Mr.
Rama.
I will open their voice for the questions. Have some questions. Kayala Mamadova and Ayten Shkirova. Here they have some questions.
Ayten Shkirova. Yeah, okay. So boss is Raman. So Raman decides.
Hello. Hello.
Yes, we can hear.
I am Ayten Şüküroğlu from Azerbaijan Medical University, Department of Pharmacology. I am senior lecturer in the department. Even I have been at Turkey University. We do our scientific work in that laboratory, Hacettepe, Anadolu. And I am glad to see your PowerPoint. It was so interesting. And I have a question about laser saliva. Which active compound you identified impact diabetes? It is so important for me because I am interested in diabetes.
I'm very sorry, I cannot hear you very clearly. Can you just repeat the last part? Last part, please.
About laser salvia.
Oh, laser. Okay.
Which active compound impact directly diabetes? And what they do? Increase or decrease glycogen level?
Okay. Actually, it is a very interesting work or compound. You know, I happen to just know this compound through the papers. You know, it is not my own work, but Dr. John Eng from States, he was able to develop these compounds just by coincidence, actually, from the saliva or venom of this creature called Gila monster. He was able to isolate some protein type of compounds that I showed the chemical structures. If you can remember, it's a very big molecule, very big, with a lot of you know, oxygen breaches and whatever. So it is called exanthem 4. It's the synthetic derivative of exenatide. It is a drug called Byetta, actually, Byetta, available in pharmacies. So it is a very complex protein. The actual protein is bigger, but they took some part of this protein and developed into exanthem 4, and you know, commercialized as an anti-diabetic drug. So from saliva to pharmacy. So this is the commercial, how it is announced. So there is a very interesting story behind discovery of this compound, but just you know, not to take longer time of the participants through this talk, I am not going to go into details, but you can find it easily from Google or from scientific papers.
Okay,
there's an interesting story behind that, how, you know, coincidentally it was discovered, like the guy, Dr. John Eng, the compound in his hand, he was just, you know, going from one company to another one to, you know, develop this compound into a commercial drug, but none of these pharmaceutical companies were not interested in, and somehow some small company found him and got this molecule, and then, you know, it got bigger and bigger and became like $1 million of drugs. But you know, in the end, he didn't take any share. But he said that when they asked this person, how do you feel, you know, the company has $1 million of sales with this drug that was discovered, but you have nothing. And he said that I don't mind, this is just a gift to human being from myself. So I think this was the, you know, the major spot of this story, but you can just go through it.
Thanks for answering my question clearly. Thanks for everything.
Thank you too for asking this question.
Thanks.
I must say you have Indian name. It is Dr. Kamla Badalova.
Mr. Raman, Kayala Mamadova want to ask something. Kayala Mamadova.
Okay,
Please.
you need to...
Hey.
Kayala. Hayala.
Yes, go ahead.
Please, Mr. Raman, can you
Okay.
her voice, Kayala Mamadova?
Okay. Go ahead with your question. Do it, we'll get back to her. So we'll, many are waiting now, so we will just see if we can connect. Vandana Patrivale is here. Dr. Ilkai, I must tell, she is one of the top researchers here from ICT Mumbai. And here you are. Question.
Sure. Great insight, Professor Ilkai, and you are coming from a beautiful place. In fact, visited when Professor Hengel retired. So, in fact, I missed the earlier part of your work because I myself was delivering a webinar. But looking at the later part, you just said that you are not working on nanoparticles, but I'm sure you may be having some idea when they are encapsulated systems. Typically, you know, cosmeceuticals are encapsulated systems because of the stability issues. Do you have any studies to indicate what is their biodistribution, especially whether they reach brain? Because that's a major concern companies here in India are raising. If we are using any micro or in fact nano specifically particles, whether they'll be reaching brain. And second part of my question is, could you throw some light on the membranes which are being used as substitutes for skin? Some which are just synthetic and some which have fibroblasts cultured, and there are many more. So these two questions, if you could answer.
Yes. So very nice to meeting you through the online conference. And anyway, the first part of your question is, yes, of course, it is very important to reach at the brain, and it is easier for small molecules to you know, pass the blood-brain barrier. Honestly, I don't have any facility to do this kind of work. Only in silico assays we can just, you know, predict how it goes. I have this kind of data, but not really in experimental like benchtop assay. But it would be very nice if you had the chance, as you can also emphasize. But for small molecules, of course, and especially for small volatile molecules, we can hope more to pass the brain to the brain. For the second part of your question, actually, yes, it is not maybe enough to do only enzyme inhibition assay, but also, as I said, we have just started to do it in cell culture assays as well. Like we have a system, is small cell culture lab in our faculty that I collaborate with a colleague from pharmaceutical toxicology department. She is very good in this kind of cell culture assays, and I just wrote a project which was accepted by the grant foundation of our university. We just started to screen the compounds. I don't know if you have another chance, maybe next time I can present you also results of cell culture assays related to skin. I have some results not done in my faculty, but we also collaborated with another chemical company which have cell culture lab. So some of our extracts and compounds were tested in there. I didn't mention in my presentation, but we tested them in Hakat cells.
Oh, okay.
Yes, I have results, and one more cell line, the fibroblasts. I forgot the name of the cell lines, but anyway, two kind of cell lines we tried, but I just didn't, you know, it didn't pop up in my mind that I could have also mentioned about those compounds. Maybe next time, I hope.
Thank you so much, Professor.
You're welcome. You're welcome. Thank you for the question. Very nice questions. Thank you.
Thank you, madam. We have one more from Mohammad Kasif. After that, I'll request the participants to mail your queries, if you have any, to Dr. Ilkai.
Yes.
So, Mohammad, can you unmute yourself? Okay. Dr. Khayala, you can have a question.
Okay. Thank you very much, Dr. Ilkai, for your impressive and very nice presentation. I want to give one question. You told about incretin mimetics, which is inhibitor, about treatment of type 2 diabetes.
Ah,
Incretin
diabetes?
mimetics, yes, which use
Okay.
treatment of type 2 diabetes.
Yeah.
I want to know that what is the mechanism of inhibitory mechanism of this compound,
Okay.
and
Yeah, go ahead, please.
and I want I know that you also told that we are doing about molecular docking studies, in silico studies. I want to know that can you know that which type of inhibitor it is? I mean that it is competitive, uncompetitive, or which type of inhibitor this one?
The first part of the work, it is about incretin mimetics. This is not my subject. This is my research area. I just wanted to mention about that a natural product, like exanthine 4 can from venom of a what do you call it, reptile, can be developed into a clinically available drug. This is a very beautiful story. Sometimes this kind of natural products can open a new drug class like incretin mimetics. So I'm not really into mechanism of this kind of mimetics. So I really wouldn't know anything about that. I just know the story, but I don't know exactly the mechanism, how they interact for diabetes, because this is not my subject, not my own scope. So coming to your second question, yes, of course, through these enzyme inhibition results, you can do further, what do you call it, KI values. Through KI values, you can just calculate if they are either competitive or non-competitive, or reversible or irreversible. I have this kind of data for some of the compounds that I presented, but I just didn't put, but of course we can do not only with in silico studies, of course we can also do it with in silico analysis, but also with this IC50 values, as you know, we can, through some, what do you call it, some graphs, we can just
Mihalis Menten graph.
figure out their competitive or non-competitive inhibitors, of course. So I should maybe put some examples, but I didn't. In my papers, but you can see.
Okay, thank you very much. Thank you very much.
Thank you too. Your daughter?
Yeah. Because there are many questions, but I know even I want to talk lots to you. But fine, I understand you. Thank you, Dr. Ilkay. Of excellence. God bless. Thank you.
I can hear you clearly, Raman. I can't hear you.
Once again, in fact, I'd like to repeat it 100 times that we are
Okay.
to Dr. Ilkay for her time and the session of excellence. God bless.
Thank you. You too. God bless you too.
Thank you, dear
So we close it? So now we are closing it?
Yes, we will close it now. We will meet back once again on Facebook and WhatsApp.
Okay. Sure.
Thank you, dear participants, for making the more useful interactive participant. You have any last word to say, Ilkay, or shall I close?
Thank you very much, all of you, for participating in my talk, and I hope you enjoyed it. I'm very happy to meet you through online talk. So I hope to meet you again, maybe face to face in some part of the world.
Yeah, definitely. And again, for the benefits of the participant, Ilkay has been to India, and to Bangalore as well. And she loved the place.
Yeah. 10 years ago. 10 years ago.
So with the hope to meet soon. Thank you.
Yes.
Bye-bye.
Thank you too. Bye-bye, everybody. Bye-bye. Bitiremiyorum. Leave diyorum, bitmiyor. Allah Allah. Neden kapatamadım? Stop video diyeyim.